Before we get started, I have a quick favor. I've been self-funding the Finding Genius podcast for 5 years now. I've done over 3,000 episodes. And as you can see on YouTube, we're up over 1 million views on the channel, which is fantastic. The next thing I really want to push on is to get up to 10,000 subscribers, because once we do, we'll be able to put a donate button, and we'll be able to solicit donations to help keep the podcast running and to also get the Finding Genius Foundation moving along. We have a big project studying anxiety, depression, and PTSD, and working on a product to help people overcome these problems, because I've seen them explode recently after the last 2 years of the whole virus situation. So if you would, please subscribe to the podcast. That would help us tremendously. Give us a thumbs up and check in the description for Buy Me a Coffee. It's about $5. If you could buy me a coffee, I'd really appreciate it. It would help keep the channel going, and I love coffee. Thank you.
Good day, everyone. My name is Maher Abou Hachem, and I'm a professor at the Technical University of Denmark, Copenhagen. My research area focuses around the discovery and applications of carbohydrate-active enzymes, and I have been focusing for more than a decade on enzymes from the human gut microbiota. And I'm very happy today to share with you information about our discovery of how we can, starting from the A and B blood, use a discovered enzyme cocktail to convert this to the universal type O. Thank you.
Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. My guest today is Maher Abou Hachem. We're talking about a really interesting article I saw how to transform, type A blood, type B blood into a universal type blood, AB positive, etc. I think it'll be a really interesting call. He's a professor at Technical University of Denmark. He's in the Department of Biotechnology and Biomedicine. So, we'll get into it now. So, Maher, thank you for coming.
I'm very delighted to be here telling you about our work.
All right. So just for people that don't know, what are the major blood types? And in terms of receiving blood transfusion or giving them, which ones can receive and donate to everybody?
Right. So there are several different blood typing systems. However, as far as transfusion medicine is concerned, and also as far as organ transplantation is concerned, the clinically most relevant system is the ABO system. And this system is basically based on complex sugar markers that are displayed on the surfaces of red blood cells. And depending on the identity of these sugars and the structures of these glycans or complex sugar markers, one can be of the A blood group, B blood group, each defined by a specific marker, and the third type would be a combination of the A and B, and that would be the AB blood group, and the fourth would be the so-called O group, which lacks both the A and B antigens. And translating this into transfusion compatibility would mean that if you're an A blood type, then you would be able to give blood to the A group and to the AB group. So you would be able to give your blood to recipients which have the A antigen as a part of their blood group markers, and thereby that would lack the antibodies against this blood group. So if you give the A or B blood types, for instance, to an O blood group, which lacks these sugar markers, recipients of this blood group would have antibodies, and that would mean that these antibodies would recognize the sugar markers, and that potentially would lead to an incompatibility, positive cross-match reaction. And that can be of different severity. The most severe type would result in the destruction of the red blood cells, and thereby it's potentially fatal to the recipient.
What would happen, let's say my blood type is B and I get a transfusion, from someone with blood type A, what would happen to me?
this could potentially be fatal because it would result in agglutination, which meaning that the blood groups, red blood cells would clump together, and that means they will ultimately be destroyed. And that means that the transfer of oxygen in the body and to the peripheral tissue, to the brain, would be impaired, and that could have fatal consequences. So therefore, matching recipient and donor groups is of great significance to maintain the safety of this medical procedure, which is needed for many different types of medical procedures that require blood transfusion.
Good. All right. So you've been able to change one blood type to another. Let's get into that process. How does that happen?
So as I mentioned previously, since the complex sugar markers that define these blood groups are carbohydrate-based, it means that they are synthesized by special enzymes that would add these sugars one at a time, resembling, for instance, building a Lego type of column. And the A and B blood group are both synthesized from the H antigen, which is the precursor that is shared by all humans. And that means that if we were able to have scissors that would cut these specific markers for the A and B blood group, then we would be able to convert those red blood cells into the precursor H antigen, which corresponds to the universal blood donor. So the universal blood donor would be able to give blood to any recipient in a safe manner without any prior matching.
So when you have A-type blood or B-type blood, what makes it A? What makes it positive or negative? I guess you could have like A positive, A negative, B positive, B negative, O positive.
Right.
So what are these two factors like specifically? What do they look like?
So when you say blood group A, it means that you have on the top of this universal type O H antigen, you have installed an N-acetyl galactosamine, the specific monosaccharide. And the positive and negative refer to another blood typing group, which is based on the presence or absence of a protein on the surface of red blood cells. But these protein markers, they are, as I understand it, less significant with regard to safety of transfusion. The most important thing is to match the ABO type markers, that is the sugar markers. And actually, the idea of enzymatic conversion of A or B blood group to the O has been coined or has been established already in 1982 by the pioneering work of people in the New York Blood Center, in fact. And they demonstrated this concept by getting an enzyme to cleave the blood group B into the H antigen. Only problem then that they did this with an enzyme from coffee beans, and this wasn't particularly efficient. So they demonstrated...
What do you mean efficient? What happened?
It means that you needed huge amounts of the enzyme to convert 1 unit of group B blood. And that was possible to do, but it wasn't a very good enzyme because it wasn't adapted to activity on whole red blood cells. And yet these people have demonstrated the concept, feasibility, and the ability to transfuse this blood into later on to other recipients. The only problem then was that although the recipients survived this and they had healthy red blood cells and other markers, there was signs of mild incompatibility. And at the time this wasn't possible to explain, and this was thought to be due to the inefficiency of the enzyme, meaning that the enzymes didn't cleave each and every sugar marker, but there was some residual B antigens that were not cleaved, and they were thought to be the reason of this incompatibility.
Why are these sugars on the outside of, the blood cells, and what's their significance? Like, how do they interact with other factors in the body? Does anyone know?
Yeah, sure. I mean, the sugars actually are recognized by non-self. So the sugars are recognized by non-self antibodies that are present, for instance, in group recipients who don't have the A and B antigens, and thereby they have the antibodies against these antigens. So if you give these persons a red blood sample with B antigens, their antibodies would bind to it, and they would bind to cells, making them clump together, making them agglutinate.
Okay.
So if you remove these sort of antigens and convert the B antigen to the O antigen, then there will be no agglutination because the antibodies will not be able to bind to these sites. So
what is there? So what differential bindings in the body? Like how different would the sites be? So if there's type A blood versus type B, what would the binding pattern look like? Or if you have A AB? Is now there double coverage of these sites? Like, why?
This is basically due to biological diversity of humans. So it's the enzymes which synthesize the A and B originated from the same precursor, and certain mutations in these enzymes lead to the synthesis of the A antigen. So they add an N-acetyl galactosamine, whereas the group B antigen is synthesized by adding a galactose. So it's very subtle differences between the A and B blood group. It's a small N-acetyl group on one of the sugars, but that has great consequences with regard to the antibody bindings. So there are some antibodies that bind to the A antigen and some bind to the B. And if you have both, if you have an enzyme which has some mutations, but it's able to accommodate and synthesize both the A and the B, then you are typed as an AB person, because you would have on your red blood cells some A antigens and some B antigens.
Before we continue, I've been personally funding the Finding Genius podcast for 4.5 years now, which has led to 2,700 plus interviews of clinicians, researchers, scientists, CEOs, and other amazing people who are working to advance science and improve our lives and our world. Even though this podcast gets 100,000 plus downloads a month, we need your help to reach hundreds of thousands more worldwide. Please visit findinggeniuspodcast.com and click on Support Us. We have 3 levels of membership from $10 to $49 a month, including perks such as the ability to see ahead in our interview calendar and ask questions of upcoming guests, transcripts of podcasts you're interested in, the ability to request specific topics or guests, and more. Visit findinggeniuspodcast.com and click Support Us today. Now back to the show.
So receiving blood of the wrong type can kill you. It's very serious. So some people that, the blood bank doesn't have the blood they need, let's say they're in a rural area or just a shortage, they could die without it. So I guess it's the critical thing to be able to do it and convert the blood to different types. So, So what does that look like? Has it successfully been done, and has it been used in clinic? Like, what stage is this at, and what are the effects appear to be of doing this?
So we just published this work, and what we did differently from the guys who pioneered this concept back in 1982 and others who followed them was that all these studies were centered on the textbook sort of paradigm that the blood group A is defined only by the A antigen, and that blood group B was defined only based on the B antigen. Okay. However, what we did different is to look into the literature, and we have recognized that there's additional antigens that are found on group A red blood cells, which are extensions of the A antigen. Again, using the Lego analogy, this means that if you are having the A antigen, that means that there's another enzyme which is adding 1 sugar or 2 sugars or 3 sugars on the top of the A antigen. And that would, of course, cap the A antigen. And that means if you try to use the A enzyme to cleave this A antigen, it will not work because there are different structures which are blocking this enzyme, right? And that would mean that this concept would be a dead end, because no matter how efficient enzymes you give, they would only be able to convert the exposed A antigen, but not all the extended forms. And there are 3 of them. And the same goes for the B antigen. There's not only the B antigen, but there's also another extended form where 1 single sugar is added on the top of that to cap it, again precluding access to the B antigen of enzymes targeting this antigen. So what we've done instead is recognizing that these structures are present, but recognizing also that they have been overlooked in the literature, and their significance to blood transfusion and particularly to the enzymatic conversion of blood has not been explored. And then what we did was to choose a specific specialist human gut symbiont, a bacterium which resides in our guts, which is specialized on targeting the main component of the mucosal layer that lines our gut cells. And This component is called mucin. And the reason we did this is because the mucin in our guts, it turns out that the A, B, O antigens are also displayed as peripheral sugars on this glycoprotein. So we simply harnessed the co-evolution of this human gut bacterium to live and to nourish itself from these mucin O-glycans, including the ABO antigens, and discovered a whole enzyme battery targeting not only the known conventional A and B antigens, but targeting all their extensions.
Is this like a complete acceptance of the new blood type then? Like, why do you have to go to this extent to do all this?
Because simply these extensions have not been recognized as a part of the A and B blood group phenotype. So they weren't recognized formally as antigens that would have antibodies in recipient and that will cause incompatibility. So all the work that people have been doing was to discover better enzymes against the A and B antigens, and that would never have led to fully compatible blood due to the reason that these other structures exist and they are antigenic, and therefore they are an integral part of the A and B phenotype. And this is what our work shows.
Instead of trying to force compatibility and block things, you're trying to do it in a more natural way that it'll be accepted more than forced upon the body. It sounds like.
Yeah, I mean, simply if we don't do this, then we are presenting some structures which we know now, based on our study for a fact, are problematic. So we have established for the first time that these extended blood groups are problematic. They will cause incompatibility if not removed. And the way we showed that is by we performed more than 1,500 compatibility tests where we treated red blood cells either with enzymes that remove the sort of canonical A and B antigens, or with our discovered enzyme blend, which not only removes the A and B antigens, but it removes also their extended structures. And the outcome of that was that we showed that our strategy resulted in a significant improvement of compatibility using the cocktail against both the canonical and extended sugars as compared to only removing the A or B antigens.
Okay. So have transfusions been done with this altered blood, and, what is the outcome versus unaltered? Have you tried it on matched blood, unmatched blood, etc.? Like, how's this been evaluated?
So the way we evaluated this is the way this is done in the clinic. So in the clinic, before you perform transfusion, then you have a gel card assay where the plasma from the recipient is included in this assay in a column, and then you add the red blood cells from the donor. And if the red blood cells are compatible with the recipient's plasma, then they will migrate through this column and reach the bottom of it when they are centrifuged. Whereas if they are not compatible, they will agglutinate, and they will form larger aggregates that are unable to migrate in the same manner. And the more retarded the cells are, the more severe this positive cross-match, and the less compatible the cells are. And since this is used in the clinic, we used the same concept to evaluate compatibility. So the answer to your question is compatibility was done during this assay, valid in the clinic, but we did not transfuse our converted blood cell into any recipients yet, because that will need to go through some additional tests and some additional safety measures, etc.
Are there any transformations that are more problematic than others? Let's say A to B is easier, or B to O, or A to O, where they all seem to be equally difficult?
That's a good question, because the answer is yes, there is a difference. So I mentioned to you that the group B blood is phenotypically simpler because it has only one extension, whereas the A blood group has 3 extensions, meaning that we are targeting 4 structures, the A and 3 of its extensions. And when we did the compatibility test, we more or less abolished incompatibility in the B red blood cells. There were anywhere from 91 to 96 fully compatible assays out of 100, and depending on the genetic background of the recipient, or of the donor rather, whereas in the A blood there was a significant number of remaining cross matches. So we had only compatibility in the order of 50% of the tests, which means that we still have some work to do there to understand the underpinning basis of this incompatibility. Whereas for the B blood group, we are we have up to 96% compatibility, and those remaining cross matches are weak. So not only our enzyme cocktails improve compatibility, but they reduce the remaining cross matches in severity. So we have milder cross reactions.
Interesting. Okay. How is this going to be tested? Like what needs to be done once you go through, more safety protocols and everything? Is this going to be tested in, is it even possible to test in mice? Is this going to be in organoids, or is it going to go straight into people? Like, how will it work?
Yeah, that's another good question. So fortunately, we've been funded for some further research into this to investigate different scenarios to explain the persistent cross matches, particularly to the A blood. And when we reach to the point where we feel we have understood this and where we have decreased the cross matches sufficiently to be able to go to testing, we will not be able to carry such experiments on mice because they don't have the same ABO system. So to use animals for this, then we need to go for higher primates. And the lowest of these higher primates is the Jipong ape, and these are kind of endangered species, so I don't think we're going to try to do that. But then the next step would be to perform tests on a limited number of humans and to perform transfusion of smaller amounts to evaluate the compatibility and whether or not there's any immune reactions and how severe they are. And if that works, then obviously this will be expanded to reach to the point where this can make it to the clinic, should everything work out.
Yeah, no, that's very interesting. I don't know if anyone knows this. What happens to a person once they get a transfusion of blood? Let's say, I don't know, I'm type AB positive and I get O positive. You could tell that that's not my blood because there's no AB markers in it. Has anyone looked somewhat longitudinally? What happens after I get a transfusion of that after like day 1, week 1, month 1? What's observed?
Yeah. So I'm a protein chemist and a molecular microbiologist. So this is not my core competence, but to what I understand it, this has been done already when they transfused the first B-derived enzymatic blood. And what they do is they monitor, for instance, they monitor the survivability of the red blood cells. So they label the red blood cells in a special way, and they are able to track how do these cells survive in the body. They are also following changes in the morphology of these in the patient. They are following an immune response from the patient. So the emergence, for instance, of antibodies in the bloodstream of the patient. So there's some amount of different tests to make sure that the cells are okay and they are surviving and they are doing their job of oxygen transfer. And on the other side, looking at from the patient's side of view, looking if the patient is in any way launching an immune response against these cells or if they are just accepted as one's own red blood cells.
Okay. All right. Well, very good. Yeah, Vahan, this is a very interesting technology. I think it's going to be amazingly beneficial once it comes. Last question, any estimate of the size of the need for blood to be available for people where it's not currently available? Like any estimate on the size of shortage or the frequency of shortage, in the U.S. or other countries?
Yeah, that's also a good question. And again, I'll have to improvise there. So from what I know, about 118 million donations are performed every year worldwide. And as the system looks now, some of the more industrialized countries, like Denmark, for instance, they have a very functional blood management system where, and they have reasonable donor pool. So essentially, at the time being, this is not a severe problem here. But in other countries, this is a challenge, and because it costs a lot of resources to qualify donor blood for transfusion. It needs to be tested for various viruses or other issues, and then the blood center or the hospitals need to have a stock of all the 4 blood types. However, the use of the universal blood is disproportionately high because this can be given at medical emergencies, it can be given to people of ambiguous blood typing, etc. So this means that there's a lot of logistics to keep this system in place, and probably this would be more problematic in rural areas or in less well-developed areas of the world. And given the average age of the population is increasing, there will be an increased demand on blood due to increasing medical procedures, cancer, you name it. So probably this type of solution will be useful to mitigate the challenge of simplifying the availability of blood simply.
Okay. Well, very good. Where can people keep tabs on the development of this matter? Where can they go?
Well, I mean, basically, as I said, we are working on this for the next 3.5 years or so at least. So people are welcome to check our websites, our lab output. But otherwise, I'm sure the news will travel. I mean, this has been a very well sort of propagated news after we published this article, because this is really something that people can relate to and they can appreciate the problem and the solution to it. So hopefully we'll communicate this also via news media and via popular science fora and via talking to people like yourself.
Well, very good. Maher, thank you so much for coming on the podcast. I really appreciate it for your good work.
Thank you very much. You're most welcome. And yeah, hopefully this will be beneficial to your listeners.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.